and welcome to another episode of Virtual MOSI. My name is Amber, and today we're going to be talking about world-renowned scientist Elizabeth Blackburn, who studied chromosomes, which are the storage units of our DNA. So let's talk a little bit more about the background before we get to her specific work. So DNA, as you may have heard of, it's a big word that stands for deoxyribonucleic acid. So sounds pretty big, right? But if you break it down, ribo just means sugar. Deoxy means without oxygen. So deoxyribose just means that the DNA molecule has a sugar that has one less oxygen in it. So let's start looking at the structure of our DNA. As you may have seen in our model so far, it's just a double helix, and that just means that straight it would look like a ladder, but it's twisted. So that's what makes it that double helix. So our backbone, which is where our sugar is that we were talking about, has sugar molecules and phosphates, and that makes the outside portion of our ladder. And then inside we have our nucleic acids, which are our bases. And the order of these bases are really important because they determine the genes that make our traits. So those traits include eye color, hair color, your height. All of these things are determined by your genetic code and the order of those bases. So if you want let's learn a little bit more about those bases and make a little model. So I'm just using candy today. You can do this at home really easy. I'm using some candy ropes, some candy drops, and toothpicks to hold it together. So some basic things you need to know to do this at home are what each of these pieces represent for our model. So our candy ropes are our backbone. That's the sugar and the phosphate that we were just talking about that goes on the outside. And then the really important part are these bases. So if you're making this at home, you'll need 4 different colors. That way you can see the different bases. Our cytosine, which we're representing as C, guanine, which represented as a G, adenine, A, and thymine, T. If you're making this at home, make up your own genetic codes. It's super fun. Just keep one rule in mind. C always binds with G, and A binds with T. So if you want to make this at home, all you need is to take a toothpick, choose your genes. So if you want to do a CG bind, all you need is the color you're using for C, the color you're using for G. Take the toothpick and carefully push it through. Remember, it's a little sharp, so try not to poke yourself, and just make a few of those bases. And the order will determines the different genes in your body, but if you're making this at home, feel free to make up your own genes for whatever order you want to put those in. So I'll make a few of these so you can kind of see what it looks like, and then we'll put it all together with our backbone so that we can see the full structure. So while you're doing this, think about how all of those bases come together and how just simple changes make so many different traits in our body. You may be 5 foot tall when you get older, but your best friend could be 6 foot tall. So what makes that person different? Well, it's all of these different genes put together to make you uniquely you. All right, so I'm just adding on that backbone right now, and you just push it through on each side. You're making just a little ladder. And like I said, this order is what determines what gene you're making. All right, let's see if we can get a few more on here so that you can see the full double helix. All right. And this one's a pretty easy one to do at home. If you want to learn more about DNA, we did also have a video back in March about DNA extraction, so you can learn more there. So now we have our ladder. That's the basic form of our DNA, but in our bodies, it's actually twisted, and that's what makes that double helix. So you can kind of see that there. The double helix is just based on the way that the molecules in here bind together and cause that structure. So make that at home, and that way you can kind of see and feel what it's like for our DNA, which is really, really tiny in our bodies, but it's really, really long. So since it's really long, it has to be stored. It's pretty important, so we don't want it to get damaged, so that storage has to make it as safe as possible. So how do we do that? Well, in our bodies, our DNA gets really, really twisted and wound up as tightly as possible, and that is what, and when that's twisted up like that, it's called a centrosome. So was anybody able to guess how many centrosomes we have in our body? No? That's all right. So in humans, we have 23 pairs, total of 46 chromosomes. And the reason why we have pairs of chromosomes is because you get one from each of your biological parents. So one from a mother and one from a father, 23 from each, making a total of 46. And that's how many chromosomes we have. All right, so let's talk a little bit more about the structure of a chromosome. So like I said, chromosomes are mostly just twisted up DNA that gets really, really, really tightly bound together using a few different other molecules. And so that we can visualize that a little bit, let's look at this little model. And all I'm using is some sculpting foam. And this works really well because it looks like almost like a bunch of little molecules together. And that's basically what chromosomes are made of. So you want to start by making two fairly long pieces like this. And these are going to be the arms of our chromosome. So you have two of those together, and that's our chromosome arms. And then chromosomes are held together. These arms are held together at some point. It could be directly in the center, could be all the way at the ends, could be all along the different places. Different chromosomes are just held at different points. So for today's model, I'm just going to connect them right here. And the point that they meet, and I'll use another piece of foam in a different color so you can kind of see that point, that is called our centromere. And our centromere is important because it holds those chromosome pieces together, and it's also important because during cell division, when all the cells in your body divide, your body needs all of these chromosomes, so it kind of helps guide it around during that process. Like I said, these part are our centrosome arms, so leave that there. And then the last portion that we're going to talk about today are our telomeres. So our telomeres are just little caps on the end of our chromosomes. And so we're going to put those in here as this blue color. And this is super fun to do at home if you just want to kind of visualize and work through all these different parts. I made these little flags out of just paper and toothpicks and put those in there. But on the ends, like I said, are our telomeres. And those are very important. So if you remember from earlier, our DNA is made up of all these different bases. And they're pretty important because they have all of those traits for all the things in our bodies. But every time we divide our cells, these chromosomes can get a little damaged, especially on the ends, because they're right on the outside, and that could cause them to get in contact with things and kind of break apart. So in order for our chromosomes and our DNA to stay safe, what happens is telomeres are little repeating segments at the end of chromosomes that are just made of those bases we were talking about earlier, but they're repeating. And so they're not important for traits, they're just protective at the ends. And so we can put that on there. See, now we have our important parts of our chromosome. We have the arms of our chromosome, and our telomeres on the end, and our centromere in the center. So now let's talk more about our renowned scientist we were talking about earlier. Her name is Elizabeth Blackburn, and she studied those telomeres that we were talking about. So she was born in Australia, and she came to study her PhD at Cambridge University in the UK. From there, she came to the United States to study at Yale. And while she was studying at Yale, she was studying a single-celled organism. She was looking at its DNA, and she saw at the end of the DNA all these repeating segments. And she thought, well, why does DNA have that? She and her colleagues later learned that those were the telomeres that protect our DNA. She also worked with them to understand how those telomeres are put on using a specific type of enzyme, and the work that they did on telomeres actually earned them a Nobel Prize in physiology and medicine. All right, well, isn't it really cool what scientists can do? Do we have any final questions today?
Randini Meyer says, hello, MOSI.
Hello, thank you for watching. It's really awesome to have you guys. All right, well, if we don't have any questions today, still feel free to put those down in the comments, and we'll be able to answer those as we go throughout the week. Thank you guys so much for joining us today, and keep discovering.